We have 4 talks today. So first of all, I would like to thank the organizing committee of Synapsa that I'm here. So thank you very much for that invitation. I'm super happy to be back here in Slovenia. And we have, I think, a very nice session with 4 speakers. Okay, I have to get back just 1 second. So we're going to talk about mobile brain-body imaging. I will start this session giving a little bit of a background and an introduction on what mobile brain-body imaging is and why we do that. And this will be followed by a presentation by Dr. Nenner, Federica Nenner, who will talk about embodiment during physical interaction with an industrial robot as one example how you can use mobile brain-body imaging to better understand human interaction with technical systems. This will be followed by Mansa Pesca's talk on detecting early signs of Parkinson's disease. I think this is an ongoing research where mobile brain-body imaging actually is used in clinical context. So this is very exciting. New insights in potentially finding biomarkers during actual movement of patients. And then Uroš, whom I have the pleasure to share this session with, will end this session with a talk on brain mapping during increased gravity gradients. Again, an extreme environment where we try, or where Uroš and his team tries to measure human brain activity to better understand actually what the brain does in natural and not so natural environments. So let me start with just a quick recap on how we ended up here today. Some, I don't know, 100 years ago, actually, claimed Berger introduced EEG, electroencephalography, for investigation of human brain activity. Several researchers before him did that already for like 25, 30 years. Adolf Beck, Roman Schipulski, using EEG in animal research actually and describing the main factors behind, for example, alpha blocking. But if you consider the size of the instrumentations that were used then, that was like pretty much have a room full of equipment, amplifier technology to record human brain activity, and this has drastically changed. If you look at this graph here, you can see the number of publications using wireless EEG in their approaches, starting around like some early 80s and 90s, basically, where you see mainly low channel numbers in sleep recordings, but also for epileptic, new approaches for tracking epilepsy and seizure foci. And then basically this dwindles around up till the late 2000s, and somewhere around 2007, 2009, you see this very steep increase. This is associated with an increase in systems that are available today, that are small, lightweight, and that allow assessment of human EEG activity in mobile participants. Emotive is one of the off-the-shelf systems that you can buy with 5 electrodes, pretty inexpensive. And this is also the reason why this is a system that is used in most of these studies. We're talking about 2,100 something studies. And then this is followed by M-Brain Train here, a neighbor located in Belgrade, Interaxon, Neurosky, and some other systems. Scarily enough, nearly 20% of these EEG systems are homemade. And also, if you look at the number of channels that are used, this is this graph down here, you can see that most of the studies here work with EEG with a pretty low channel range. Nonetheless, these data impressively show that EEG can be used in mobile context. So mobile EEG is possible, and the main fields that these kind of systems are applied to are cognitive monitoring, from attention to sleep, to mental load assessment, to clinical practice, where you have a number of applications up to communication with devices, so brain-computer interfacing and other things. For mobile EEG, it is not so important to basically understand how the brain controls behavior or how behavior influences brain. This is related to the field that we're talking about today, mainly mobile brain body imaging. So it's not only mobile EEG, but it's trying to add additional modalities to understand how the human brain is influenced by behavior and how it controls active behavior. So I'm coming from an embodied cognition perspective, and I try to understand how embodiment works and how our brain basically realizes embodied action in a dynamically changing environment. And I'm going to give you a brief introduction of why we think this is important and show you then 2 examples. Just as a short technical note, we planned the symposium so that all the 4 speakers basically present their contents and maybe 1 short question after each of the talks, and then we have a final discussion in the end of the symposium. But if there's like any questions related to content understanding, please just interrupt me at any time. So if we think about active behavior, then basically we can see, or as Ware and Dickinson coined it in their work, that the neuroarchitecture, so the hardwiring between neurons, the network itself does not change drastically or instantaneously when you basically start actively behaving, but the connectivity within existing neural networks changes drastically. And that changes immediately the moment that you start acting or behaving actively as compared to resting. And this makes a lot of sense. It's pretty clear that if you move, you will have vestibular feedback about the acceleration of your body in space. You will have proprioceptive feedback about your muscles and joints moving in space. And you will have motor commands. All that differs drastically from what we have done over the last decades in trying to understand how the human brain supports cognitive processes. What we do is we record people lying supine in a scanner, not allowed to move at all, or sitting in front of a monitor in light-controlled experimental labs. And then we basically record millions of bits of information from brain dynamics during these tasks, and we try to compare this high-dimensional space of information about brain activity with 1 bit of activity that is usually a button press at the end of the trial. And this is not what our brain has evolved to. Arguably, the brain evolved to optimize the outcome of our behavior, and our behavior is adapted to a dynamically changing environment. We are embodied and acted in an environment, basically, to make sense of our perception. And we use our physical structure to reach cognitive goals. So this is not natural brain activity as we would like to better understand it using mobile brain body imaging. The question then is, how do you then investigate a mobile brain? And this is pretty straightforward. We just came from the number of new systems that are available on the market, EEG systems, and obviously that is the first that we need. We need some device to measure human brain activity. This has to be mobile, lightweight, tetherless, basically, so that participants are able to move around in space. And there are tons of systems. EEG, besides functional near-infrared spectroscopy, are the only 2 systems at the moment. I'm pretty sure you all heard about optically pumped MEG. That might be one next solution in the future. People are working on mobile PET systems, but this is talk of the future. So if we want to understand human brain dynamics in behavior and how that relates to active behavior, we have to synchronize behavioral recordings with EEG activity. And that is usually done with motion capture. In addition to that, we use very often head-mounted virtual reality to have immersed participants, but still very highly controlled experimental protocols. And on top of that, you can obviously add everything that is available, including eye tracking in the new head-mounted VR system and peripheral physiology to get a more complex, holistic, if you like to say that, insight into human physiological responses during active behaviors. So this is what we do, and this is part of our lab that you can see here in the background. So this is one of our standard setups. And obviously, this is a non-trivial task to solve, having wireless systems that come in with different sample rates, high data packages basically being transferred that have to be synchronized. There are architectures out there. Lab streaming layer is one of these architectures. It's open access, so everybody can use that. It's an open community architecture where everybody can contribute and write drivers for new hardware to be incorporated, for example. So this part is already solved, and I hope there will be better solutions in the future. But then there's also other issues. Data-driven analysis methods are necessary if we have active behavior. And this example, I'm not going to dive into that, but what you can see here is like two simulated sources in the left motor cortex and the right parietal cortex oscillating at different alpha frequencies, and this has slowed down a realistic head model. This is the inverse solution. We know the sources and project that forward due to volume conduction. What you can see is that the activity of these two sources can be measured everywhere as a linear mixture of all sources being active in parallel. And this is only volume conduction. We're not talking about eye movement, facial and neck musculature that contribute to capacitive conduction to every single channel that we record. So we need different analysis methods to dissociate brain from non-brain activity and then subsequently use better and realistic head models to estimate the real sources of their origin. This would be one example where we now included eyes and facial muscles plus neck musculature into a realistic forward model for better source reconstruction using mobile brain-body imaging approaches. So these are the technical details, and I don't want to bore you too much with that, but I would like to give you two examples that for me are at the very core of mobile brain-body imaging, where we try to understand how behavior influences brain activity. And both examples come from spatial cognition research, which makes a lot of sense because vestibular input and proprioceptive input are pretty important sensory sources for spatial updating. And the first example here is heading computation, with the vestibular system assumed to contribute significantly through acceleration and velocity signals to the updating of heading in space. And what we did here was a simple setup. Not technically, this is the setup. So you can see again, we use a Zotac high-tech gaming computer basically in a backpack that has like a motion capture calibrated head-mounted VR system. In this case, we used 156 actively amplified channels with like 28 channels on the neck to get additional neck muscle activity for a better reconstruction of the sources. And the task was pretty simple. Participants would stand in the middle of our lab, and they would see, this is a bird's eye view, basically they would see an orienting beacon at the beginning of each trial. That would be replaced by a sphere that then started moving with two different velocity profiles around participants and ended unpredictably on the trial between 30 and 150 degrees plus filler trials to both directions, left and right. Then it would change its color, and that was the imperative stimulus for participants, basically to rotate back and indicate their initial heading. And with that kind of response, we then get everything that we need. Behaviorally, we have the acceleration, we have velocity during the rotation, outward and backward rotation. We have over and underestimation of the behavior itself, how basically they judge, participants judge their heading. And crucially, what we do, and we try to do that as often as possible, we compare a traditional laboratory setup that you can see on the right side here. That is the visual flow, the exact same visual flow that people would see, but using a joystick to follow the sphere. So this would replicate what we know from our traditional lab setups. And then we compare that with the head-mounted virtual reality setup, the MoBI setup here. So we would call that the stationary setup, and this is a MoBI setup. The visual flow would be pretty much the same, but the movement obviously differs drastically. If you look at the behavior, what you can see is an increasing, more or less monotonic increasing absolute heading error as a function of the eccentricity of the end position that you can see here on the x-axis. Y-axis is the heading error in degrees, and this is in blue for the stationary setup. Now for the MoBI setup, and this is the behavior, this is the basis for all our interpretations. What you can see is that when participants have the full range of natural idiothetic senses during this outward rotation, their heading estimate is significantly better. And you see no increase up to around 90 to 100 degrees, and then there's an increase basically when you cross your cardinal body axis, 90 degrees to either side, there seems to be a switch in the spatial representation to respond to this cognitive task. Importantly now, the question is, how do we get brain dynamics and behavior together? And we started, this is a long project, and I try to give you a tour de force of like 5 years trying to find an analytical approach to get to this data. What we did is we used spatial filtering approaches. In this case, this is independent component analysis, and you can see each of these little dots here, the spheres, are one participant, one independent component of one participant, using a k-means clustering in multidimensional space of these EEG activities of the ICs, so the biggest sphere here is the center of that. What you can see here is that this cluster is located not really in the brain, but more or less outside the brain, likely reflecting muscle activity, that of the sternocleidomastoid. That would be attached to the mastoid and then to the sternum, and that basically is activated when you rotate your head inward and down. So whenever you have a rotation of the head, this muscle would be activated. What we then did is we computed the spectrogram of the full outward rotation for all the different frequencies, and we chunked the outward rotation according to the velocity during the rotation. And we sorted the velocity according to the lowest 10% to the highest 10% velocity trials, and sorted accordingly the frequencies. So it's a Hilbert transform band power for filtered frequencies that are sorted according to the velocity during the outward rotation, 10% lowest to 10% highest. And what you can see here then is basically if you have a striping pattern, then you can see that for the different frequencies here, from 3 to 5, non-overlapping up to 30 hertz, 3 to 30 hertz. And if you see a striping pattern within each of these frequency bars here, you would say basically that velocity has an impact on the power or vice versa. This is the first step, and we then basically computed for all these different frequencies and all different velocity bins, a 180 times 180 dissimilarity matrix, using basically representational dissimilarity approaches, and computed the distance in multidimensional space here. So this, using Mahalanobis distance, is the same as Euclidean distance for higher dimensional spaces. And then we basically show the differences between the different conditions. First of all, again, it's sorted like the way before, but now we compute the differences for the upper triangle here, only the MoBI condition. So we look at differences in different frequency bands dependent on the velocity of the outward rotation, within MoBI only, between MoBI and the stationary setup, and between the stationary setup, or within the stationary setup only. What you can see then is basically doing the statistics, permutation statistics in this case, and then plot only what is significant. And this is the real data of the right cluster representing neck muscle activity. What you see is a velocity-dependent modulation of these neck muscles that are visible only in the physical rotation, but not in the joystick rotation. You can see tons of differences, velocity-dependent, in the MoBI condition and between MoBI and joystick, but there's nothing in the joystick condition. And that is also the result when you do the statistics. That makes a lot of sense. If you're using a joystick to control the rotation, your head is not moving. There is no muscle activity. You're still standing in front of the screen, but you're moving with the joystick. But obviously, velocity modulates neck muscle activity. This is a proof of concept that this kind of approach works analytically. Now the question is, and I'm going to show you two examples. One that was super surprising to us, that is the occipital cortex. That's a cluster in or near occipital cortex, and obviously it's EEG. We have no precise estimation of the source, but it's an estimation, and we're talking about a range of 1 centimeter to 2 centimeters, which can be a lot, you all know that. But this is more or less all within striatal and extrastriatal areas, and you see not much of a velocity-dependent modulation for the physical rotation, but lots of activation for the joystick condition. If you do the dissimilarity and the statistics, there is no activity in visual cortex during an active physical rotation to follow a sphere. And this fits perfectly with predictive processing. This is, or in other terms like Mittelstadt and Mittelstadt, reafference principle. Von Holz and Mittelstadt, if I compute an action, I send out a copy of the afferences that will come back during execution of the action. If that fits, both of these signals basically nil out, and the residual is movement in the outside world. And this is what we can see here. And this makes this is super impressive and was surprising, something that we've never seen, and bears a question whether all these alpha modulations that we see in tons of EEG experiments where you have visual motion on the screen but no physical motion might be associated with sensory mismatch processing rather than the cognitive process we are trying to investigate. Another example would be the retrosplenial cortex, but I'm not going into that. This is just an example that shows, yes, of course, visual information is sufficient to reach specific cognitive goals, and you can update heading based on visual information, but it's just not as good as if you have the full information. Okay. This was the first example, and I think a very good example to show the relation between active behavior and brain activity. And the second is a newer study that hopefully will be published in the next days, where we worked with 12 patients that got their right hippocampus entirely removed, benign tumors, epileptic patients mainly. And the question was, what would happen if we have right hippocampal resectomy patients doing the classical Morris water maze? Again, this is, I don't know whether you all know the task, this is a classical task for spatial diagnostics in hippocampus patients, where you have to use allocentric or global landmarks basically to triangulate your position and the position of a target to find in this kind of circular arena that has no geometric information to indicate where something would be located. So we used different scenes here, alternated through that, and we had 12 patients with the right hippocampus entirely removed, 2 age-matched entire health control groups. And this is one of the examples. It's kind of a little bit slow. This is one of the patients, basically, again, doing the Morris water maze on the stationary setup here. So in the desktop, you can see the lab, and she was placed in the lab doing the entire thing on the desktop, and then the same task was done again with head-mounted virtual reality, allowing patients and healthy controls to move actively in space. And then obviously, the question is, what would happen to patients and controls between the two different conditions? What you see here is just a short insight in some of the parameters that we computed. This is a spatial memory score, where basically we distributed thousands of potential stops randomly in this arena and computed the percentage of your location and the likelihood of that being not outside or one of the random distributions. So the higher the score here, the better. What you see here on the left side is a stationary condition, on the right the mobile condition. Then you see in blue the medial temporal lobe patients, and in orange the control patients. What you can see is here is in the stationary condition, patients are much worse than healthy controls in a standard diagnostic tool for hippocampal resectomy. That's not very surprising. What is surprising is that if patients are allowed to actively move, there is no difference between patients and controls anymore. Patients are just as good, even though the entire right hippocampus is gone. They are just as good as healthy controls. And this clearly indicates that there are extra hippocampal strategies, sensory information that is used to solve this task that cannot be done based on visual flow information only. The same is true for the scatter of the final location, basically asking whether the final location was placed randomly, which is not the case here. The scores are inverse. If you look at basically the likelihood or the response location percentage here, you can see that from the stationary condition on the left-hand side to the mobile condition, there is an increase, a concentration of the likelihood of being in the target location that improves again from the stationary to the mobile condition. And just an example of one patient which shows a very typical pattern. This is the probe trial. We had 4 probe trials, so you learn the location of the target in 3 trials, and then there are 4 probe trials where the starting location now is rotated, basically requiring you to use external global landmarks to update position and triangulate the target location. And this participant basically in the desktop condition, this is the identical first probe trial, same starting location, but now the starting location changes, and what she does is simply replicating the exact same path. This is a pure egocentric strategy. It's just a path replication that does not make use of any of the landmarks at all. And she does that basically in all 4 locations. If you look at the MoBI condition, however, things change drastically, and the strategy is entirely swapped. She basically would move to the center of the arena, look out for landmarks, and then directly return to the target location, and she does that from all starting locations. So there is a drastic change in behavior. The question is, can that be observed also in the brain dynamics? And this is, again, just presence probability in these different arenas between the 2 conditions. You see basically less entropy, the more mobile, the more idiosyncratic information you get. Okay, and this is just a short summary, the data analysis ongoing. I'm super excited about this, and one of the aspects that we are focusing on at the moment is gamma activity. What you can see here is like the beginning of the start, first 2 seconds, 3 seconds in the middle of each trial, and then the last 2 seconds before the target is indicated at a cluster of frontal midline sensors now. We're not talking about source space, but sensor space at the moment. And this is healthy controls, and this is the medial temporal lobe patients. You see slight differences in theta. I'm not going to discuss that. What is more impressive if you now look at the difference between stationary and mobile. And what you see in the mobile condition is a massive gamma burst. Everybody working in this field would immediately say, yeah, that's an artifact, that's movement related, it's like muscle, cable, or whatever related to movement. This is after removing all non-brain sources, after spatial filtering, plus the baseline that we are subtracting here is an active movement baseline without navigation goal. So the movement is exact same in the baseline as it is in the task, so it's very unlikely that this is a pure artifact. Nonetheless, the more important point is here that this gamma activity is massively pronounced in healthy participants, but not so much in patients, and you can plot that in different ways. You can look at the spatial distribution in veridical space, basically, so you just plot gamma power as a function of location in that space without considering the distance to the target or the rotating starting locations. Then you can see in the stationary condition, again here, the medial temporal lobe patients, on the right-hand side, the control. In the stationary for gamma, 30 to 60 hertz, and this is the mobile condition. Again, you see a drastic change in gamma activity in healthy controls only, not so much in patients, and this is not the same all over the place, basically, again, arguing against a pure movement-related artifact. This would be a target-centered space, so we just rotate the distance of those spots with reference to the target location. The same pattern here, stationary, and a massive increase in healthy controls in the mobile condition. If you rearrange that and now rotate the different starting locations in the probe trials, what you can see here, only MoBI, again, a massive difference in the healthy controls in the gamma range. So, as I said, we are not yet there to make any very strong conclusions in this study, but I'm very excited about this data. So, summarizing, if we start moving, if we investigate brain activity in actively behaving participants, obviously, and by definition, due to movement-related sensory feedback that will be processed by the brain, brain dynamics will differ from stationary setups that we have used for the last decades to understand human brain activity. So it is not surprising. If you then move into the real world, which several groups have done, we do that too. We look at spatial orientation behavior using EEG and multimodal recordings. Here, this is an example of navigation systems in Berlin. then basically you lose control. You get additional information into your data that has to be separated from what is actually of interest to you. And as a summary, then basically it is clear that behavioral states of participants influence the brain dynamic state, and that we have to address that systematically in upcoming studies. And as a side note, the Krakow paper, which I think makes a very valid point here, requires and also points to the fact that we need dedicated and fine-scale, fine-grained described behavioral approaches to understand what the brain actually does. Without behavior, we cannot interpret anything that's going on in the brain. And mobile brain-body imaging, from my perspective, obviously that's biased, but I hope you can agree, provides new insights into human brain activity that accompany natural behavior, and that is different from what we know. And finally, that this kind of approach complements and extends decades of traditional imaging approaches and leaves entirely new avenues also for clinical research, specifically movement-related disorders, Parkinson disease, and several clinical areas that can benefit from that. With that, I would like to thank all our collaborators. And one last point I have to make, obviously next year, if you're interested, here in Slovenia, we will have the MoBI conference in Piran, beautiful location, and tons of people only working in this field. So you're all invited.